So before going for any next or new topic, we'll discuss a little bit about few reactors which you have not covered in the last few classes. And these kind of reactors are actually used in different bioprocesses. So one of the common type of wastewater treatment process is activated sludge process. And in bioprocesses also we have seen CSTR with cell recycle. So activated sludge process is nothing but CSTR with cell recycle. Sorry. So in CSTR with cell recycle, you have one vessel, and then whatever cell we generate, that should be recycled back to the system. So we take the biomass and put back to the system and maintain desired concentration. So here we actually operate the system continuously by supplying air, and feed will be added to the system, and similarly continuously certain amount of treated effluent will go through the outlet of the settler, and this is your waste sludge. Now large scale systems are okay, means this ASP is good for using them in a very large community, and wherever you have wastewater strength significantly low, means if you have really high strength wastewater, you will go for anaerobic treatment. Now in small volume system, or wherever you need lower capacity system, there you can actually have a different kind of configuration that's called sequential or sequencing batch reactor. So here actually we'll have multiple batch reactors, or this single batch reactor also you can have if you just operate in a given sequence. So you will get total 5 different stages. So first is called filling or fill the reactor. Second is the reaction. So you try to fill the reactor. So essentially you are doing this operation in a batch mode. So fill the reactor with raw material or your wastewater or reactant. Then you have reaction stage, that's called reaction. So in the same reactor, so I'm just talking about the stages of operation. So you have actually dealing with single reactor, but different reaction stages are there. So here you will have actually, suppose you need to provide air. So you do aeration. If you need to mix the liquid by with the help of external recirculation, then you can have recirculation by pump, and you do all your reaction in this stage. And after finishing the reaction, the third stage is called settling of biomasses. So in this third stage, you just settle the, you just allow the system to settle down. So whatever bacteria grown in the reaction stage, they will try to settle down in the third stage, and fourth stage is actually yours. So after settling, the solid biomass will try to create a different layer, and from top you will get the clear or liquid having less biomass will be there on the top layer, and then you can do removal of the product. So here it is called decant or removal. And then finally, you will have a system which is idle. So the idle stage is actually preparatory stage for the next batch. So in the sequencing batch reactor, you are actually essentially maintaining multiple environmental conditions in a single batch reactor, which is not very common when you use normal batch reactor where you put everything and then just wait for the batch time to process the reactant, and then finally you just remove this, open the system and remove the product. Whereas in sequencing batch reactor, even if you go for say 2 different system, or maybe if you have 5 such batch reactor running in series, then also you can do this operation in a continuous mode. Why we are calling it in continuous mode? Because if you just draw a bigger box having all reactor inside, then essentially from this box, if you do the input output, if you try to make the input output diagram, so first fill, so you have certain amount of time you need to spend for filling, then the liquid will go for reaction. So all reactor you will just fill. Now we'll go for reaction. Then after reaction, you will just have.
Sir?
Yes.
Sir, can I just quickly ask one question? My internet connection is going on and off, so I just wanted to ask one question. Sir, about the minor. So, sir, there are 6 people here in the campus.
No.
So, can they also give offline, online?
No. In the campus, as per Dean's office record, on 8th February, the number was 14.
No, sir.
No, I have the record. I have the record. I got the record for Dean Student Affairs Office, and as per the record, 14 students are there, and I can actually name the student also. The 14 students are there in the campus. Now, today also they have sent another updated data sheet, and that is considering the onboarded student data till 10th February.
Yes, sir.
So I have already asked ERP to provide me the course-wise data, so I'll get the data soon. So whoever will stay in the campus, they have to come to LHC and appear in the exam in the classroom, in the classroom. And, and rest of the student will actually give the exam in online mode. So we'll have both. Yes.
This is based on today's list, right?
Yes, I'll try to follow that. Means obviously it will take some time for me to. It is not only for one course. I have to do it for all 600 courses. So yes.
Okay, sir.
So will it be a open book exam?
No. So even for online also it will be proctored. So the we'll create multiple MS Team channels, and in MS Team channels we'll have means different TA will be proctored the exam rooms, and those will not turn on their camera or they will not follow the instruction, will not consider their exam paper for evaluation, or they have to come to the, whenever they will come to the campus, they will reappear in the exam. So if you say that your internet connectivity is poor, you couldn't turn on the camera or you couldn't follow the instruction, all are okay, because we are quite sure that before major exam we'll bring back the student to the campus. So in that case, the student who will not be able to follow the instruction or will not be able to turn on the camera during the exam, they have to reappear in the minor exam. So I'll conduct another minor exam for them and they have to reappear in the classroom.
Yes, sir. Sir, so do we have to take laptops to the LHC or will it be like
No,
written?
pen paper. Laptop is not required for LSC because issue with LSC is in all rooms we don't have sufficient number of flag points. So even if you bring your laptop and if you don't have sufficient charge in the laptop, that can create problem.
Yes, sir. So so we will have a written paper then for those giving offline because you said it was going to be on Google Forms.
Yeah. So I'll create the same form and print it. So you have to just fill in the blanks. So you have to write very long answers because the way we are setting the paper, only few words you have to write. So you will get sufficient amount of time, whether it is on online mode or in pen paper mode. You will barely have any difference. There also you have to read the question on screen. Similarly, here also in hard copy you have to read the question. So it will not take much. It means the time difference will not be very long.
Okay, sir. Okay.
Insignificant. Okay. So in sequencing batch reactor, you have option to do this operation. If you have, suppose you're taking two batch reactors. So when one is in filling condition, other one can actually have the next. So when you do this operation, first you fill the first reactor and also the second reactor as well. Or maybe first you don't fill the first reactor, first you fill the second reactor. And then when it will go for reaction step, that time you fill this. So whenever this will go for reaction, the second reactor will actually follow the third stage, that is settling stage. So when first reactor will follow settling stage, the second reactor will actually follow the removal stage. And when it will follow removal stage, it will actually go to the idle stage. That means preparation for the next batch. So in a sense, you add something and then so if you just have these 5 reactors in a particular box, and if you start them in a sequential manner when you start the operation, so first you fill this. When it will go to reaction stage, you fill the fourth one. When it will go to reaction stage, fill the third one. So in that way, if you just do it, then actually you can operate the entire scheme in a continuous mode because one of the other reactor will be empty, which can receive the fresh feed. So although your operation will be in batch mode, but essentially when you check the bigger scheme, like if you put everything in a single box, then you will have only one input and one output from the bigger box.
So are these like 5 different reactors or it's just the same reactor and all the reactions are happening sequentially?
So same, you can actually have single reactor, then you have to wait for all different times. Another is if you just run this batch reactors in sequence, so multiple similar configuration of reactor, but multiple numbers, then you can actually operate them in such a way that overall scheme will show like a continuous operation.
So these are 5 different reactors, right?
Yes, you can. Yes, you can have that if you really want to have a continuous type of operation. And each reactor will experience all 5 stages, like filling, reaction, settling, and removal plus preparation for the next batch. So advantage is, as I told you, that in small scale system, wherever the quantity of reactant or quantity of wastewater is less, their sequencing batch reactor can be used for one major advantage, that is you don't need a separate settler. You also don't need to provide oxygen continuously compared to the other configuration. ASP, you can continuously supply air, and for settling tank, you just get the settled biomass, put it back to the system, and continuously you get the clean water from the settler. So this is a continuous mode of operation where continuously you're supplying air. Now you do this because if you need to have this bigger size reactor in multiple number, then maintenance of that so many reactors will be very, very expensive. Whereas if your working volume is less, then you can still manage with this sequencing batch reactor. Reason is the first reactor, and if you can remember in your wastewater treatment, we have also mentioned that initially you will have one settler to remove different other component that is part of primary or upstream operation. So here also in the reactor, as it is a continuous operation, you always maintain a specific volume in the reactor. So volume is constant here. So you are not getting any change in volume. Similarly, in this system, first you fill the reactor. So you are just trying to achieve the desired volume. To a certain number. And then you aerate. So you aerate only when you talk about reaction. You are not running the aeration continuously for entire period. So after aeration, after reaction period, you stop aeration. Then you allow the biomass to settle down. And after settling, then rest of things are actually like any other mechanical separation operation, where you try to separate things based on their settling velocity. And here we are talking about gravity separation, means we are not taking any help of induced gravity like centrifugation. So it will settle down and then liquid can be removed from the top. So advantage is when you try to manage lower volume system, then you can have multiple smaller reactor. And in treatment plant, essentially you have chambers. So when you go to any treatment plant where they have sequencing batch reactor, they will have chambers separated by wall. So you just imagine a long box where you have just multiple separator to get individual chambers. So one chamber to another chamber, it is very easy to move then. And you also can have option for changing the environment. Like here, suppose we are using two different reactor system. One will be operated in presence of oxygen. Another will be operated in absence of oxygen. So you run the system in presence of oxygen to remove, say, carbon, and also oxidize to, so carbon will get oxidized to carbon dioxide. Ammonia will get converted to nitrate, and phosphate will stay a little bit utilized by the bacteria, but phosphate will stay. Most of the phosphate will stay here. Now in the second stage, the nitrate, because it is anaerobic now, nitrate will get converted to nitrogen, and the phosphate will be utilized by phosphate accumulating bacteria, or that's called phosphate accumulating organism. There, this phosphate accumulating organism, they prefer to get exposed to both the environment, aerobic as well as anaerobic. So whenever they will get exposed to anaerobic condition, they will try to take up the phosphate and store them inside the cell. And whenever they will go to aerobic environment, they will try to release the phosphate to the environment. So sometimes for phosphate removal, you deliberately put the same bacteria in two different conditions, aerobic and anaerobic, to get rid of phosphate from the liquid system. So this is about your sequencing batch reactor. And another reactor system is also widely used for low volume and low strength wastewater. So wherever you have low strength and also sometimes low volume, then you can have a tower type reactor. Inside the tower you have packing material. So biofilm will grow on the surface of the packing material, and from top you'll just pour the water at a specific flow rate, and from bottom you will just collect the treated water. And it can be used for any other chemical reaction also. So from top you will put the reactant, or here it is wastewater, and from bottom you will get the treated water. And water will actually just trickle down the bed as water is flowing through this channel and trickling down the bed and getting collected at the bottom portion for getting the treated water. This kind of configuration is typically in chemical engineering, we call them as packed bed reactor. So it is packed bed reactor. And in packed bed reactor, when you just have the counter current movement of air and water, so for this kind of reactor system, air will actually move from bottom to top and liquid will come from the top. So you will have a counter current movement of water and air. So in some system you can have external air source, air supply line, and some system you will not have any air supply line, only you will have some holes at the bottom in such a way that water will not leak, but air will pass through the bed. So when you supply air, then it is called active air supply or active aeration, or when you don't supply any active air, then it's called passive aeration or passively aerated system. So one of the common design for treatment plant is trickling filter. It looks like filter because this solid media containing biofilm there on the top, they look like a filter media, and as water is trickling down through this bed, that's why it is known as trickling filter. Advantage is you are not supplying, and most of the cases we actually work with passively aerated system because most of the strength is less. So the residence time in the system is maintained in such a way that air water contact will be sufficient enough to treat the waste, and also you will get the trickling behavior of water. So we call them as trickling filter, and it is also one example of passively aerated system. And when you use such kind of system, obviously treatment cost will be reduced. So low volume, low strength wastewater can be treated using this system. Same reactor can be used for other biochemical process also. When we will discuss about acetic acid, vinegar, acetic acid production, that time we can see the use of similar kind of reactor. So this is also another kind of reactor system which we have not discussed earlier. So sequencing batch reactor and trickling filter, these two reactor systems are quite well known as well as widely used for handling mostly low volume or low strength wastewater. Now we'll discuss about a specific process which has a relation between bioprocess, conventional bioprocess or high value bioprocess system, and wastewater treatment. So that's called bioethanol production. So ethanol can be synthesized using chemical means, but biologically, if you try to synthesize ethanol, you have multiple options. You can choose different food substrate, like different grains you can choose, and which commonly used for production of different beverages. Like in beer industry, they use, say, barley, corn, rice. So different food material you can use to produce or to get ethanol out of those food material using fermentation. So when you try to do that, then you get substantial amount of ethanol, but essentially you are sacrificing food material. So when we talk about ethanol production industrially, we have their usage for different industrial purposes. So whatever ethanol we produce using fermentation from different food material, they are mostly consumed as alcoholic beverages. Wherever you need to use ethanol for any other industrial purposes, either you chemically synthesize them or you use some other cheap materials to produce ethanol from those cheap material using biological means. Now, ethanol is also has good ethanol also has good ignition properties, as well as it can be used for fuel substitutes, and many places to improve the or reduce the greenhouse gas emission, or improve the fuel efficiency, or improve the green fuel efficiency rather, people try to blend gasoline or petrol with ethanol, and then you can actually have little bit economical operation of engines. Means transport, obviously when you reduce the fuel price, it will have significant impact in other sector also. So in India, there is a target to have 20% blending of ethanol in alcohol, petrol. So to get this higher amount of ethanol, because 20% ethanol in petrol is quite significant. In India, if you talk about petrol usage, so in a given day we use huge amount of petrol, and to compensate 20% of that demand, you need significant amount of ethanol production. So chemically, you can try to synthesize ethanol, which may cost you per liter around 30 rupees, say, per liter. Now, when you use biological means, your target is to use different kind of waste material to produce ethanol. So when you use this ethanol as a fuel substitute, then also you call it as a source of fuel. So when you produce ethanol directly from food substrate, it's called first generation bioethanol production, because you are directly using some food substrate to produce ethanol. Now, when you move towards some waste material like lignocellulosic material, then you talk about second generation bioethanol production. So first generation is relying on food substrate like barley, corn, rice, different food substitutes. Whereas second generation is actually not relying on food, it is relying on some other waste residue, and you do little bit of chemical or biochemical treatment prior to ethanol production, because in second generation ethanol production, we try to use biomass. So second generation, we try to use biomass, and then we do pre-treatment. And finally, after pre-treatment, we try to hydrolyze, and after hydrolysis, we do the fermentation. And then we have purification for getting sufficient concentration. So most of the biomass, when you talk about biomass, we mostly talk about different lignocellulosic residues. Like if you use wood chips or any other waste biomasses like rice straw, wheat straw, or different grasses, even sugarcane bagasse, means different sugarcane residues. So all this material has significant amount of cellulosic substance. So our intention is to get the cellulose, because cellulose is a good carbon source. So get the cellulose from the biomass, and after getting the cellulose, we'll hydrolyze the cellulose to get the monomers like different sugars, mostly 6 carbon and 5 carbon sugars, pentose and hexose. And then we use some organism which will use this pentose and hexose sugars to produce ethanol. And after getting the ethanol, we just do purification, because if you get say 5% or 6% ethanol in the broth, you need to separate that and up concentrate that to get 100% water-free ethanol before you blending it to the petrol for ignition purpose. So the entire process is cheaper except this stage. So when you try to compare the cost effectiveness with any chemical processes, the entire process, fermentation, hydrolysis, there, and purification is actually common for chemical as well as biological process. There also they use distillation. Here also you need to use distillation. So entire process is cheaper because biochemical fermentation would not require too much of energy. Hydrolysis is also not require too much of energy. Only pre-treatment is quite expensive. As of now, people just use different expensive pre-treatment method to get rid of lignin. So when you talk about cellulosic material present in the biomass, cellulosic material is actually not present in the biomass in exposed form. So mostly you will have a layer of or protective layer of lignin around the cellulosic biomass. So the lignocellulosic, that's why we call them as lignocellulosic biomass. So the biomass that will have different cellulosic fibers layer, and all these fibers layers will be covered or protected by lignin. And this lignin is it is really very tough substrate to break using biological methods also, or sorry, chemical methods also. So typically either steam explosion or acid treatment, alkali treatment, or high temperature treatments are done to get rid of lignin. And when you do this kind of treatment process, you also lose certain amount of cellulosic fractions. So losing of cellulose is essentially reduce the carbon content available for fermentation. So you will lose certain carbon when you use very harsh chemical treatment. And also in chemical treatment process, sometimes they will produce some residues which are not very good for the bacteria or yeast present in the fermentation system. So suppose it is producing some other acids. So whenever you have production of other acids during chemical treatment, that other acid will actually create inhibition. And different biological treatment processes are also tried. And in IIT Delhi, we also have developed a biological treatment system which can remove the lignin without degrading cellulose. So we are not going to waste cellulose, and the system can effectively remove lignin. Only difference is steam explosion or size reduction. So for lignin removal, you can have treatment with steam, treatment with acid, treatment with alkali, treatment with treatment or size reduction. When you try to reduce the size, means if you just chop the biomass at a very fine size, then also you will have significant breakage of this protective layer of lignin. So size reduction, and then you have biological treatment or enzymatic treatment. So when you use enzymatic treatment using any biological means, mostly we rely on fungi which can secrete ligninase. So a mixture of enzyme which will degrade the lignin, and only the problem with this system is they are inherently slow. you need substantial amount of biomass, and also you are dealing with solid state fermentation, because in liquid media it is a little bit easier. Contact time will be less because it will get mostly homogeneous system. Whereas in solid state fermentation, you need to ensure that sufficient amount of contact is there between two different solid material, and enzyme secreted by this. If you get fungi which has a tendency to secrete enzyme, like if you have production of extracellular enzyme, then it will be more beneficial. So in our lab, we have actually isolated a strain which can secrete this enzyme, and with the help of that enzyme, you can do the treatment of lignosulfuric material. Only problem is the treatment time required for this is almost 10 to 20 times higher compared to rest of the treatment options. Advantage is you are not losing any residues, and also you are not producing any other inhibitors. Whereas here it is producing inhibitors, and it is also, you will also lose some cellulosic residues. But you can achieve the you can complete the entire operation in 1 to 2 hours. Whereas here it will take almost 1.5 days to 3 days. So it is a little bit longer, but only advantage is, and for this operation also, the operation is very simple. You have to just soak the material in a tank and then keep it there for 1 and 2 days, around 2 days time, and you get good removal. So when you try to compare, it is slow, but it is more cost effective. You are not adding any external chemicals, and also you are not creating any high temperature, high pressure environment. So cost of treatment is less, but it is more time consuming. Now, if you have substantial reduction in cost in this treatment process, then overall cost will also get reduced. And obviously you have to match with this cost. If your production cost is actually exceeding this, then people will not show much interest, because they will say that if you do go for chemical synthesis, you can get much cheaper. It means you can get ethanol at a much cheaper rate. So obviously will not go for this kind of system. But now, because of different advanced enzyme also, people are looking at the ethanol production from biomasses. Now in the second stage, whenever you will have hydrolysis, there also you need to use certain enzymes which will help you to hydrolyze the biomass, and after hydrolysis you'll end up by getting pentose and hexose sugar, which can be fermented by different kind of organism, like Saccharomyces cerevisiae is there, Pichia stipitis is there, so even Gymnomonas mobilis is there. So bacteria or fungi you can use to produce ethanol from the pentose and hexose sugars. So you can have simultaneous saccharification, and so this hydrolysis is also known as saccharification. So you can have simultaneous hydrolysis and fermentation single reactor, or you can have in two different reactors. And for fermentation also, you can have sequential fermentation, like fermentation done by 5-carbon utilizing organism, fermentation done by, then followed by fermentation of 6-carbon organism, or vice versa. Or you can have organism which has capability to utilize both 5-carbon and 6-carbon both and produce ethanol. And in fermentation, mostly we prefer to get organism which can utilize both the carbons effectively, 5, like pentose sugar and hexose sugar, and also they should have higher ethanol tolerance limit. So if your organism has a tolerance limit of say 10% or 15% ethanol in the broth, then actually you can do the fermentation a little bit for a longer period, and you can finally you can get a high concentration of ethanol in the broth, and subsequently that ethanol, the broth can be moved through centrifugation or filtration to separate the biomass, and the liquid can be again sent to the distillation column, and in the distillation column you get the azeotropic mixture, and just to break azeotrope, you can add different other reagent. And one of the common reagent is say toluene. So you can add, break the azeotrope, and then finally you can get the 100% ethanol through azeotropic distillation. So this is one of the scheme for production of second generation bioethanol or second generation bioethanol, which can be used as a fuel substitute. And there, most expensive treatment step is pre-treatment. And to make the process more economic, you have to improve the economics of pre-treatment, and also you need to make sure the fermentation can take care of both hexose and pentose, and also it can this organism should have a little bit of high tolerance to alcohol. Otherwise, the organism will actually experience product inhibition. Then you can get reasonably reasonable amount of ethanol at a competitive rate. So this is all about the course content we have covered till now is actually the syllabus for your minor and exam. If you have any queries, I'll just try to answer the questions then. So if you see the comparison between cost of ethanol operation when you use different kind of options like steam, acid, alkali, size reduction, or even after steam, you can actually have superheated steam as well as hot water. So even after steam, you can actually have another option called hot water treatment. So when you use this kind of system, then the cost, you can have a cost comparison. Just when you use steam, 1 liter ethanol would cost you almost, means for this, considering rest of the bioprocess also, will cost you almost 1 point, means it is about 80 rupees. And then when you go for rest of the process, it will be around 60 to 65 rupees. Whereas when you go for enzymatic, this particular treatment, whatever we have developed, that will cost you about 5 rupees per liter. So it is 80 rupees per liter, 6 to 65 per liter, and it is 5 rupees per liter. So obviously you can see significant reduction. Only thing is treatment time is high. Whereas in other cases, you can process the system, material at a faster rate.
So isn't the treatment time can be reduced by artificially synthesizing the ligninase enzyme and then treating the substrate into a reactor by stirring it into a aqueous medium so that efficient so it no longer remain solid fermentation.
Agreed. Actually, Novozymes is trying. So they have given one of their cocktail to test whether they are getting significant removal. So we had a research collaboration with Novozymes. I'm talking about a situation where we did this particular experiment 4 years back. That time the efficiency was not very great. And also major problem is when you try to use really very expensive enzyme, you have to recover and reuse the enzyme, then only you will have good process economy. Otherwise, every fresh batch, if require more fresh enzyme, it will be very difficult to manage. And the organism we have isolated, the novelty with that organism is whenever you take this biomass, obviously agricultural residues, means rice straw, wheat straw, or say wood chips, they will have a lot of other bacteria and fungi on the surface because we are not autoclaving them, we are not sterilizing them. So in the reactor, whenever you try to put water and suppose you are just soaking the rice straw, obviously the bacteria as well as fungi present in this on the surface of the rice straw will also try to grow if they get a little bit of substrate. And when you use enzyme, externally sourced enzyme, you have to make sure that enzyme is not getting degraded or not getting used up by the organism present in the system. And also you should get or you should recover the enzyme to a certain purity level which can be used in the next batch. Then only you can have process economy, means economic operation. Now the strain we have isolated, that actually has a very good property. Whenever it will grow, it will automatically outgrow rest of the strains. So even if you have some inherent bacteria, inherent fungi present in the this biomass, they will not be able to grow at a significant level. So the strain we have developed, isolated, that can, whenever it will grow in the system, it will automatically outcompete other strain, and then they will start degrading lignin. So when they're trying to develop this enzyme, they're also trying to make sure that this enzyme should not be, means you don't need very high pure enzyme here, you should not use, and also you should have effective reuse of enzyme to improve the process economy. So it is still under R&D stage, and they're trying to develop different enzymes. And one of the good thing is in China, the type of fermentation or operation we did, they also did similar kind of operation, and they already established the method at a larger scale. So yes, it is possible, and that's why people are actually trying to do more research in that area to improve the pre-treatment, economic feasibility of pre-treatment process, to have better end value or to have more competitive price of final product obtained from the system, bioprocess system.
Okay.
Okay. Any other question? Okay, then means we'll have the exam on, I think, Thursday, because that's the last day of a minor exam for this time. So those who are present in the hostel, they have to come to the LSC and appear in the exam, and rest of the student will actually appear in the online mode, and it will be a proctored exam. So make sure you have good connectivity, and you should have option to keep your video on for the entire examination period. So we'll have 1 hour or maybe 1.5 hours exam, but during that 1 hour or 1.5 hours long period, you make sure that your internet connectivity is okay, as well as your video connectivity is also appropriate. Otherwise, only one thing is that you have to come back and then reappear in the exam. And for assignment, I'll just share a link I'll just send it to the group. So you can just submit your assignment by Sunday night. So you'll get 2 days after the exam. Then you can submit it by Sunday night. Okay. Any other questions?
Not currently, sir.
Okay. Thank you. Thank you then. Have a good time. Okay. Best of luck for your exams, and see you after the exam, because next week there is no class. Friday also it is no teaching day. So we'll meet again next week, Tuesday.
Sure, sir. Thank you, sir.
Thank you.
Thank you.
Thank you, sir.
Thank you, sir.
Thank you.